Now, moving ahead. The single greatest predictor of a healthy gut microbiome is the diversity of plant in one's diet. Today, we have the privilege of diving into this intricate world of gut microbiome and its profound impact on wellness and illness. And to guide us, we have an expert in microbiology and research, Professor Dr. D.S. Murthy.
Dr. Murthy sir, academic journey is nothing short of inspiring. An MBBS from Usmania Medical College, an MD in microbiology from Gandhi Medical College, and advanced training from Harvard Medical School. With over 15 publications in esteemed journal, including the British medical journals, his contribution to medical microbiology are widely recognized. Currently serving as a professor of microbiology and vice principal at Sri Venkateshwara Medical College, Dr. Murthy sir has been instrumental advancing research and education in microbiology, particularly and understanding the role of microbes in human health.
Without further ado, let's welcome Professor Dr. D.S. Murthy to enlighten us with his expertise.
Let's give him a big round of applause.
Good afternoon, sir. Dr. Murthy, sir, you can start your session, sir. ఇటు పక్కన ఈ కెమెరా బాగుంటుంది, ఇటు కెమెరా పెడితే బాగుంటుంది, అటెండ్ బెటర్ అగైన్.
Good morning, all. Am I audible, sir, now?
Yes, sir, you're audible, sir.
Okay, sir. Thank you very much for such a nice introduction. And I thank Santakumari madam for inviting me here and all the organizers. As a preliminary discussion about gut-brain axis is already over, I'll be focusing primarily on the role of microbes in the gut-brain axis in the wellness and illness. Next slide, sir. So gut feelings, many times we say gut feelings. Gut feelings, are they real? And can bacteria and gut decide our health and mental status? Can bacteria in our gut influence our mood, cognition, and behavior? Can bacteria in our gut control the outcome or pathogenesis of neurological conditions like Alzheimer's disease? All these factors we'll be learning in the next 25 or 30 minutes. Next slide, sir. Next slide, right. You all know what is serotonin. What is serotonin? We call it as a happy hormone, right? So serotonin is nothing but 5-hydroxytryptamine. It is a monoamine neurotransmitter, plays several roles in the body, including our happiness, memory, learning, and also it regulates the body temperature, sleep, and sometimes sexual behavior and hunger as well. Next slide, sir. But the low levels of serotonin are associated with depression and other mood problems, anxiety, sleep problems, digestive problems, suicidal behavior, OCD, post-traumatic stress disorders, panic disorders, schizophrenia, phobias, all these things. These are all well-established facts. Now, do you know where the serotonin is produced? Next slide, sir. 90% of the serotonin is produced in the gut, in the cells lining the GI tract. Next slide, sir. Only 10% is produced in the brain, and it is released into blood circulation and absorbed by the platelets from the gut primarily. Next slide, sir. In Bhagavad Gita, there is a saying that Yuktahara viharasya yukta chestasya karmasu yukta swapnava bhodasya yogo bhavati dukkaha. That means what we take inside, either into our mind or into our body through food, will influence our happiness. Next slide, sir. You should know that there are more number of microbes than our own cells in our body. There are approximately 100 trillion cells. That means 10 times more than the number of human cells are there in our body. Just think over. Are bacteria living inside us, or we are living in the home of the bacteria? This is actually an intricate question. And the collective genome of these organisms is 100 times larger than our own genome. 90% of this lies in the intestinal tract. And of this, more than 75% is from the colon. Next slide, sir. Man, we are not just human. We can be called as super organism. Taken the amount of the bacterial load we are having, we can be called as super organism. I will introduce a few terms before I go ahead with further. Metagenomics is the study of all DNA from a given environment, which for humans includes the DNA from host as well as the microbiota. Then what is microbiota? This microbiota refers to the totality of the microorganisms that coexist within the host or in a specified part of the host's body, such as the GI tract. For example, if they are in the gut, we can call them as gut microbiota. This gut microbiota, whatever the organ they lie, they are essentially an organ. That is an essential organ. Next slide, sir. Now, there is small difference between microbiome and microbiota. Microbiome is actually a metagenome, the collective genome of all members of the microbiota in a given habitat. All microbes we can call as microbiota, and the collective genome of all these is called a microbiome. Of course, both the effect of both is same, but a small difference in the terminology. And the proteins encoded in the human intestinal microbiome, these will reflect the functions required by the intestinal bacteria to cope within the gut environment. You know, the gut environment is harsh, so it is not actually what is suitable for most organisms. So they have to survive in the harsh environment of the gut. At one extreme, there is a low pH. At the other part, there is a high pH. At all these areas, they should be able to survive. Next slide, sir. Dysbiosis is an imbalance of the microorganisms in the body, particularly in the gut. It can occur when there is too few beneficial bacteria or too many potentially harmful bacteria. That means the balance is lost. There is always a balance between harmful and useful bacteria. If the harmful bacteria overgrow or the useful bacteria become less in number, so that becomes a dysbiosis. Next slide, sir. This dysbiosis we are discussing now, but it is not new. It was observed several hundreds of years back by Anton van Leeuwenhoek in his own stool sample. Next, click next. In 1681, he first observed that disturbed microbial composition of the diarrhea he experienced. He demonstrated altered microbial flora, though he doesn't know what they are, in his own stool. Next slide, sir. I don't discuss all this, but this is technically an approach to study the gut microbiome. We can go to the next slide, sir. Next slide, sir. We call, next slide, sir. It is not changing here. Right. So we call all these things as omics, genomics, transcriptomics, metabolomics, and bioinformatics. All these tools will help us to study the vast array of microorganisms that are lying in our body. Actually, we cannot count even the number of species that are there in our body. That is a problem, but we can solve the problem to some extent by using all these bioinformatic tools. Next slide, sir. Now, I will just briefly tell a small number of organisms, the species of organisms that are present in our various parts of the gut. In the small intestine, 10 to the power of 4 to 10 to the power of 8 per ml of intestinal content will be by bacteria. Firmicutes, there is a group of bacteria, Firmicutes, that includes Streptococcus, Lactobacillus. We all know about Lactobacillus, and Coprococcus, Megaphira, Brevibacillus, Veillonella, Gemellia, and Bacteroidetes, Porphyromonas, Prevotilla, Actinobacteria, Fusobacteria, Proteobacteria. All these are different groups of bacteria that are present in the stomach. There is slight alteration in the stomach. There is 10 to the power of 3 in the stomach, whereas 10 to the power of 4 to 10 to the power of 8 per ml in the intestinal content. Next slide, sir. Next slides. Right. But in the colon and feces, contrast to the pH in the stomach. pH in the stomach is very less, but in the colon and feces, the pH is more. There is a bacterial density is also more, 10 to the power of 8 to 10 to the power of 11 per ml. And there is also transit time is long. there is 35 hours, more than 35 hours transit time. And colonic bacteria have more time to proliferate. So increased density of the contents also will be there because the water absorption is taking place in the colon. There are more than 100 bacterial species and 60 fungal species, and viruses we cannot count beyond our understanding. Next slide, sir. The important groups of bacteria that are present in the colon will be Firmicutes, Bacteroidetes, Fusobacteria, Proteobacteria, Microbiome, and Actinobacteria. These are the different groups of bacteria that are present in the colon. Don't worry about all these genus or species. In the next slides, I will focus more on the important ones that can actually influence our mind. Next slide, sir. So what they feed in the gut? The bacteria feed upon various things that are present in the gut. For example, resistant starch, undigested starch from our food, cellulose, pectins, oligosaccharides, and sugar alcohols like sorbitol and xylitol, secondary plant metabolites like flavonoids, tannins, lignins. There are some proteins like sarcoplasmic and myofibrillar proteins, some endogenous substrates like glycoproteins like mucus, hyaluronate, chondroitin sulfate, proteins like digestive enzymes, discoimed epithelial cells, bile salts. By seeing this, you should understand how much help they are doing. They're actually trying to digest the things that we cannot disintegrate in our body by using our own enzymes. And also, they're scavenging the epithelial cells, discoimed epithelial cells, and other digestive enzymes, and they're recycling them actually. Next slide, sir. The establishment of gut microbiota happens in the neonatal period itself. First exposure to the viable bacteria happens through maternal vaginal mucosa. And in the early postnatal period, there is low diversity and high inter-individual variation. But over the age grows, the diversity will increase to some extent, then it stabilizes. There are some factors like important factors will be mode of delivery and diet, especially breastfeeding versus formula feeding. Next slide. You can see the important species that establish in the gut by the time the adulthood is reached is Firmicutes, Bacteroides, Actinobacteria, Proteobacteria. These are the ones that will establish as a major groups in the gut microbiota. Next slide. The factors that will influence will be, as I said already, the breastfeeding versus formula feeding, and also when you are starting the solid foods, or are you using more antibiotics in the childhood, even the genetic factors, geographical factors, siblings, pets in the home, and vaginal delivery versus cesarean section, all these factors that will influence the microbiome in the developing in the gut of the neonate. So this is the reason why you should avoid using the antibiotics as much as possible in the childhood. Next slide. Suppose if the delivery is through vagina, the gut flora will be more amount of lactobacillus, bifidobacterium, bacteroides, and prevotilla. These are all helpful bacteria that will influence positively the overall health of the individual. Whereas if it happens through cesarean delivery, there is a less amount of lactobacillus, bifidobacterium is also less, bacteroides is also less. There is a large amount of corynebacterium. So these will favor towards disease-prone conditions. These will also influenced by exposure to the antibiotics in the early childhood. Next slide. So actually, this is the reason why you advocate normal vaginal delivery as much as possible, followed by early breastfeeding as much as possible. See, normal vaginal delivery and normal breastfeeding will favor the growth of beneficial bacteria such as bifidobacterium in the baby. There is a less number of pathogens, and there is also a pathway called intramammary pathway for bacterial transfer, in addition to the provision of prebiotics in the breast milk. Previously, I think you heard about the breast milk is one of the prebiotic, and there is, in addition to the prebiotic action of the breast milk, there is a pathway called intramammary pathway, and decreased rate of numerous chronic diseases ranging from allergic diseases to obesity is noted in the child who has breastfed. Next slide, sir. So this is a picture showing the intramammary pathway. What happens is the bacteria from the gut will be taken up by reticular endothelial cells, and they eventually land up in the lymphatics and can reach the breast milk and reach the establish in the neonate. That pathway is called enteromammary pathway. Next slide, sir. The normal functions of the gut microbiota will be, I think by this time you might have guessed many functions of this gut microbiota. Initially, I mentioned that they are removing all the harmful metabolites and also scavenging functions. So it provides intestinal barrier function, immune regulation, metabolism. They will influence the metabolism and adiposity, brain and behavioral effects, tissue and organ homeostasis. Of all these, next slides will be focusing primarily on what are the effects that can be seen in the brain. Next slide, sir. These we can call as gut feelings, that is gut-brain axis. That is a bidirectional axis. Next slide, sir. This is a bidirectional communication network linking GI tract and the central nervous system. The microbiome plays a critical role in both the wellness and disease within the system. Several metabolites produced by the gut microbiota can travel through the blood and impact the brain, and also vagus nerve plays a role. Next slide, sir. So this bidirectional communication can be through circulation. That means the metabolites from the gut can reach the brain through blood circulation, and also several impulses can pass through vagus nerve. So this is actually a bidirectional communication. Next slide. So several metabolites will be indole, from which again the serotonin can be produced, immunomodulatory mediators like sphingolipids, uropeptides, and PAMPs. PAMPs means the pathogen-associated molecular patterns, lipopolysaccharides, certain toxins that can provide our inflammatory responses, and bile salts, and certain short-chain fatty acids, and bioactive compounds, enterocytes, and all these things can be absorbed into our bloodstream. Next slide. The microbiota gut-brain neuroendocrine axis is primarily through hypothalamic pituitary adrenal axis. And the bidirectional communication is also participated by vagus nerve. Sometimes physiological stress response. I think suppose if you are anticipating in a situation, you might feel that some stress in your stomach. You are feeling stress in your brain, but it is reflecting as a stress in your stomach. So it is all mediated by vagus nerve. There are several hormones also play a role, like norepinephrine, gamma aminobutyric acid, receptor ligands, serotonin, and all these things. Next slide. So animal models establish relationship between the HPA axis and neurodegenerative diseases like Alzheimer's, Parkinson's disease, irritable bowel syndrome, and comorbid anxiety disorders. These animal models are very similar to humans, like they are mammalian models only. So we can see that this can also play a role in humans as well. Next slide, sir. So this plays a role in the regulating neurotransmitters. We'll see one by one. Reducing inflammation, enhancing the metabolism, modulating the stress and anxiety responses, and supports the cognitive functions. Next slide. For example, the key bacteria that play a role in the gut health and mind health will be Lactobacillus, Bifidobacterium, Akkermansia. These are all good bacteria versus the bad bacteria that are Clostridium and Enterobacteriaceae. These are harmful if they are in excess. Next slide, sir. Bifidobacterium, this is a bacterium that is known for anti-inflammatory properties and can produce certain neurotransmitters. Lactobacillus is known for its ability to produce certain antimicrobial peptides and regulates the immune system. Akkermansia mucinophila, this is known for its ability to produce anti-inflammatory compounds and regulates the gut barrier. It limits how much amount of metabolites that may be absorbed through gut into the blood. That is called gut-blood barrier. Cyclobacter, it can produce certain neurotransmitters that can regulate the gut-brain axis. Next slide. The neurotransmitters that are released by the gut bacteria will be serotonin, GABA, dopamine, glutamate, and norepinephrine. Need not to say the role of all these things to you. I think you know well about the role of these hormones in the brain. Around 90% of the serotonin is produced by gut, certain bacteria like Lactobacillus and Bifidobacterium. As I said already, it can regulate the mood, sleep, appetite, and several behavior patterns. And GABA helps, that is produced from the lactobacillus rhamnosus, that helps to, as a primary neurotransmitter, inhibitory neurotransmitter, reducing the anxiety and stress. Dopamine, it can affect the motivation and reward processing by the brain. Glutamate and norepinephrine, these can help in the cognition and mood stability. Next slide. These are all produced by the gut bacteria. Certain short-chain fatty acids also produced by the gut microbiome as a part of their metabolism, and they can influence the brain, especially butyrate, propionate, and acetate. These are the products from the dietary fiber. These can help to produce the short-chain fatty acids and these can reduce the neuroinflammation and strengthen the blood-brain barrier. If the blood-brain barrier is strengthened, you know that several microbes that can infect the brain will be prevented from entering the brain. So it can support the neuroplasticity and also memory functions. These fatty acids can regulate the hypothalamic, pituitary, adrenal axis functions as well and control the stress response of the humans. Next slide, sir. This gut-brain axis can also enhance the metabolism, like microbial metabolites like short-chain fatty acids, as we discussed previously, can improve the overall gut health, reduce stress response, and support brain function, and modulate stress and anxiety responses, and also support cognitive function, like even memory, learning, and neuroplasticity. Next slide, sir. Dysbiosis is an imbalance in the structure and or the function of the microbiota that leads to disruption of the host microorganism homeostasis. In simple words, the useful bacteria are becoming less in number and harmful bacteria are growing more in number. For example, Clostridium difficile, it is associated with the diarrhea and colitis. There is imbalance between commensals and Clostridium difficile. It follows over antibiotic usage. If you use antibiotics for a longer time, the normal bacteria will die and the bacteria like Clostridium difficile will selectively proliferate in the gut and can lead to actually certain conditions called pseudomembranous colitis and even can precipitate severe sepsis. In cholera and HIV also, the altered microbiota is noted. Next slide, sir. So the dysbiosis or the altered gut microbiome that plays a role in atherosclerosis, metabolic disorders like type 1 and type 2 diabetes, and other metabolic syndromes, autism, IBD, allergic diseases, and asthma. Next slide, sir. So this is one important factor here. You can see the picture clearly. That is called leaky gut syndrome. In the leaky gut syndrome, the metabolites produced by the bacteria will actually enhance the permeability of the gut epithelium, and several metabolites can pass through to reach the blood. So unnecessary or useless or dangerous chemicals, metabolites can access the gut because of the altered microbial flora in the gut. That is called leaky gut syndrome. Increased gut permeability due to the dysbiosis. Next slide. Sorry. This dysbiosis can play a role in the depression and anxiety. The lower lactobacillus and bifidobacterium, as I mentioned already, have increased pathogenic bacteria like Enterobacteriaceae, sometimes even Clostridia, and play a role in the depressive behaviors in the mice. So there are several similar studies are showing in the humans as well, but they're in the experimental stage. Probiotic supplementation can reduce the anxiety and improve the mood. Next slide, sir. Autism spectrum disorder, ASD. This ASD patients often have a gut dysbiosis. This is actually cause and effect studies are still going on, but more association is found between altered microbiota and these conditions. Increased Clostridium species is found in the ASD. And in these patients, there is a benefit with fecal microbiota transplantation. The fecal microbiota, how it is transferred is the normal persons or the healthy humans fecal matter is kept in a gelatin capsule and given to the patient. So that is improving the overall health in these patients. So that is called fecal microbiota transplantation. Next slide. Schizophrenia. Higher gut inflammation and altered microbial composition is also noted in the schizophrenia. And basically, it is caused by short chain fatty acids and microbial metabolites. Those are influencing the dopamine pathways and playing a role in the psychosis. Next slide. Alzheimer's disease and Parkinson's disease. Here also, in the Alzheimer's disease, there is an increased bacteroides, but of course, there is a decreased short chain fatty acid producing bacteria, which is contributing to the neuroinflammation and building up of the amyloid plaques. In the Parkinson's disease, there is reduced levels of the beneficial bacteria like bifidobacterium, and there is overgrowth of Enterobacteriaceae and fewer short chain fatty acid producing bacteria. That is leading to increased motor symptoms. Next slide. You can see in this picture, there are several disease conditions associated with the gut dysbiosis, especially autism. We already discussed some psychiatric disorders, pneumonias, neurodegenerative conditions, atherosclerosis, thrombosis, heart failure. All these things are influenced, even lung cancer. Some metabolites can be acting as cofactors for development of the lung cancer. And type 2 diabetes, obesity, mitochondrial dysfunction, cancer, several other cancers, skin cancer, all these things can be modulated by dysbiosis. That is because several metabolites of the microbiota can leak into the bloodstream because of leaky gut syndrome and can influence or can work as cofactors for development of all these conditions. Next slide, sir. Now, the question is, by knowing all these things, can we improve the mental health by modifying the microbiome? I think the previous speaker already mentioned about the role of the probiotics and psychobiotics as well as the prebiotics. The lactobacillus rhamnosus from the probiotics can reduce the anxiety, and we can improve our food by taking high fiber foods, for example, bananas, garlic, onions, and these will boost the beneficial gut microbiome. And fermentative foods, like for example, yogurt, kimchi, kefir. Kefir is again like almost like buttermilk, but with some other probiotic supplementation. And all these things can maintain the gut balance. Diet rich in the fiber and polyphenols, supporting a healthy microbiome. And if you want to go to therapeutically, fecal microbiota transplants are being tried for several conditions, and also lifestyle interventions. For example, exercise, sleep, stress reduction like meditation and yoga, they can positively impact the gut health. Don't underestimate all these things, because I think the theme of the CME itself is what we are is what we consume. So these are all the things we are consuming. So when we alter the foods we are consuming, we can positively influence the gut microbiome. And when the gut microbiome is towards the beneficial side, the metabolites of the gut microbiome also will be helping our immune system as well as our overall health. Next slide, sir. Meditation and microbiome, is there a link? The stress management or stress reducing techniques such as meditation, yoga can help to regulate the gut-brain axis and promote a healthy microbiome. And the reverse is also true. For example, many times we plan to do early morning walk to reduce our obesity or whatever for maintaining the good health. How many people really sustain morning walk? Some people can be motivated to continue it. Some people cannot continue it beyond 2 days or 3 days. They will stop by 4th day or the next week. And they start again after 1 month or so. Why this is happening? The motivation to do so is also influenced by the gut microbiome. There is an interesting study recently published in the British Medical Journal stating that how the gut microbiome can regulate or influence our decisions towards good habits. So please make a note of it. Next slide, sir. Fecal microbial transplantation, I already mentioned. Stool is harvested from a healthy donor that is kept in the gelatin capsule, and it can be taken orally or per rectum also. It can be introduced in the intestines so that the healthy bacteria will proliferate in the gut and replace the pathogenic bacteria in the patient. The healthy bacteria are taken from the healthy donor, and they are given as in fecal, there's some gelatin capsule to the patient. So it is supposed to correct the dysbiosis. Next slide, sir. Probiotics, I'll just add a list of probiotics. These are the live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. These are all probiotics. Next slide, sir. I will not go to the details because I think it is covered in the previous session, but this is the big list. Bacterial genera most commonly used will be Lactobacillus, Bifidobacterium, Escherichia, Enterococcus, Bacillus, Streptococcus, and Saccharomyces boulardii. All these things are given as probiotics. Next slide, sir. There are some natural products, as I already mentioned, yogurt, kefir, pickled cucumber is rich in vitamin K. Traditional buttermilk, leftover liquid from making butter, you all know this. This is actually a good probiotic. But remember, the cultured buttermilk from the supermarkets and cultured curd from the supermarkets generally does not have probiotic effect. This you should understand. Prebiotics. These are non-digestible food ingredients that confer benefits in the host. Probiotics are actually you are giving the bacteria, but the prebiotics are non-digestible food ingredients that confer the benefits on the host by stimulating the growth of bacteria. I think in the third or fourth slide, I showed that the gut bacteria require certain metabolites for their metabolism. You are providing them as prebiotics. Next slide, sir. Natural prebiotic, as we already know, is breast milk. So breastfeeding is a goal of standard infant nutrition. Next slide. Before I conclude, I want to re-emphasize what is mentioned in the Bhagavad Gita. That is, it is mentioned that there is some food like Rajasahara, Tamasahara, Satvikahara. That means the types of food what we take will influence our mood and our behaviors. Satvika, Rajasa, and Tamasa. Tamasa is like laziness, Rajasa is more aggressive, and Satvika means a calmness. So these are the types of foods that can influence that are categorized as Rajasahara, Tamasahara, Satvikahara, like that. Okay. So that means that you should understand what we take inside can influence our gut microbes and can influence our overall health. Thank you. With this, I will conclude. Thank you very much. Next slide, sir. Thank you.
We often overlook the invincible world inside us, yet it holds immense power over our health. Thank you, Dr. Murthy, sir, for unraveling the mystery of microbiomes and its profound influence on the gut-brain axis. Your insights have been both eye-opening and thought-provoking. We sincerely appreciate your time and expertise.
The speaker is now open to any questions from the audience. So anybody who has a query, you may please come forward.
Good afternoon, sir.
Yeah.
It's a very wonderful session, sir. It's quite
Thank you very much.
informative, sir, and highly in detail, sir. So I have a doubt, sir. How does exercise influence gut, sir?
How does
exercise influence gut, sir? Gut bacteria.
see, right? These are all still in the experimental stage I mentioned already. There is a positive correlation only.
Yes,
What
sir.
we know so far is based on certain diffusible metabolites and the gut analysis of the gut microbiome in the people who exercise and who don't exercise. Suppose if you exercise and have a healthy gut, the microbiome is towards the positive side. If you don't do exercise and tend to take the wrong foods, the gut microbiome is towards a different side. There is altered gut microbiome in both kinds of people. These are all called correlation studies. At this point of time, because the number of bacteria involved so many, and the number of metabolites are also involved so many, we cannot pinpoint a particular metabolite or a particular bacterium, but all these studies are based on correlation, positive correlation between the findings.
Okay, sir. So I have one more question, sir.
Right, sir.
Yes, sir. So overuse of antibiotics causes loss of gut flora, no, sir?
Correct.
if you are doing fecal transplantation, is it restoring the whole gut flora, sir?
Right,
That
it is. It can restore the gut flora, but fecal microbial transplantation has some other limitations. Unless
sir.
there is therapeutic indication like a disease process, for example, pseudomembranous colitis, we cannot try fecal microbial transplantation because the feces of the healthy carrier is also not completely characterized. We don't know what exactly we are transferring into the patient. So till that is completely, that information is available, we certainly limit this fecal microbial transplantation to certain disease indications only, not for a general metabolic corrections.
Yes, sir. Sir, is it advisable for fecal biomatter transmission through oral or any other methods? Which way
Yeah,
is
right.
more effective, sir?
I mentioned already. I think my voice became scratchy at points because of my cough,
Yes, sir.
but till we can try by both methods. Gelatin capsules can be introduced orally, and also they can be introduced through rectum also. The ultimate, they will be dissolved in intestines only. Even if you take orally, that is enteric coated, that will not be released in the stomach. That has to reach the intestines by either oral cavity or through rectum. But the second method is unpleasant. The first method is actually not very pleasing also. But the patient can or doctor can choose the method they like. But these are all enteric coated gelatin capsules containing the fecal material.
Thank you, sir.
Both methods are available.
So thank you, Dr. Murthy garu, my dear
Ma-
friend.
Madam, thank you very much, madam. Thank you very
So
much.
after a long time we could meet.
Yes,
I
madam.
have requested you immediately. You have said yes. So that
Yes,
speaks
madam.
volumes, sir. Thank
Yes,
you very
madam.
much.
That points
Excellent,
I had to go very fast, madam, because I know the time is going beyond.
excellent speech. And it shows how much connected we are still, though we
Yes, madam.
don't speak frequently. Thank you
Yes,
very
madam.
much. God
Thank you
bless
very much.
you. God
Thank you
bless
very much.
our friendship.
Thank you very much, madam. Thank you.
Can we conclude this session or any more questions from audience side? With that, we conclude this engaging session. A heartfelt thank you, sir, for your time and your making today's session interactive one.
Thank you, madam.
After an enriching session, it's time to take short musical break and refresh our minds. Music has unique way of connecting with our emotions, just like the gut and brain share an invisible connection. To elevate the energy in the room, we have a talented Shravani who will mesmerize us with her soulful voice. ब्रह्म परात्पर रामा कालात्मक परमेश्वर रामा शुद्ध ब्रह्म परात्पर रामा कालात्मक परमेश्वर रामा शीशतल्प सुख निद्रित राम ब्रह्माद्यमरा प्रार्थित राम शीशतल्प सुख निद्रित राम ब्रह्माद्यमरा प्रार्थित राम 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 जय राजा राम 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 जय सीता राम 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 जय राजा राम 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 जय सीता प्रिय गुह विनिवेदित पद राम शबरी दत्त फलाशय रामा प्रिय गुह विनिवेदित पद राम शबरी दत्त फलाशय राम हनुमत सेवित निज पद राम सीता प्राणाधारक राम राम राम
So that concludes the morning session. After a morning filled with thought-provoking insights, it's time now to nourish both our minds and bodies. So let's take a break to enjoy lunch and report back by 1:40 p.m. Thank you. By 2 o'clock, sorry. Let's report back by 2 o'clock.
music लगाना